So in this video, I'm going to start talking about how viruses replicate. So we know the structure of the different kinds of viruses, and so the next thing to look at is how do they get into the host cell and then make copies of themselves? Because we know they don't fulfill the requirements of life because they can't make their own copies. They are parasites. So this slide right here shows all of the steps, and I'm going to kind of go through these in detail. So after we're done, you can kind of come back and look through these 6 steps and sort of understand the big picture. But let's start with one thing at a time. So the first thing that must happen is we must adsorb or attach to the host cell. So we have 2 examples. In this picture we have an enveloped virus, and in this picture we have a non-enveloped virus. An enveloped virus has these spikes, which remember came from the host cell membrane. So they can very easily attach and recognize the receptor sites on the surface of the host cell. Now, non-enveloped viruses, they typically have some protein spike of some sort that allows it to be recognized by the host cell membrane as well. Now, what this does is it allows what we call a host range, that not all viruses can infect every kind of host. They need to be recognized by the host in order to infect it. So a host range could be within a certain type of organism. So this virus only affects pigs, or this virus only affects humans. But it also can be within a type of cell. So sometimes the virus could infect liver cells, but it could infect maybe cardiac muscle cells. So it's both the type of cell, but also the type of host as well that gives it that host range. Now, after the virus has attached to the proper host, now it needs to penetrate into that host. So this happens in 2 different ways based on whether or not there is an enveloped or a non-enveloped virus. If you're a non-enveloped virus, it's a process called endocytosis, in which the host cell creates a vesicle and engulfs with the membrane the virus. So you can see here it's non-enveloped, it's attached, the membrane surrounds, and you've created this little vesicle that the virus is now in. And what then needs to happen is somehow that needs to be released. The DNA needs to be released so the copying can happen. And that's the next step. We'll get to that in a minute. Now, if you're an enveloped virus, you already have this membrane that was part of the host cell you just came from. So you go over to the host cell next to you, and you very easily fuse with the membrane. And what happens is that your envelope starts to incorporate into the membrane. So you can see here that the envelope is the blue thing here, and you can see that it starts to just become part of the membrane, which then releases the nucleocapsid directly into the cytoplasm of the cell. So it's not wrapped in a little vesicle when it gets to that point. So there's some advantages or disadvantages to that evolutionary development of an envelope. Alright, so after you have adhered and now you've gotten inside, now you need to uncoat. So either enzymes within that virus vesicle or enzymes within the cytoplasm will make the envelope and the nucleocapsid break apart so that the nucleic acid is released. Once the nucleic acid is released, there is a process called synthesis, where the viral nucleic acids take control of the host's machinery and the host metabolic process and the host enzymes, and then start to make copies of their nucleic acid. If it's a DNA virus, remember that it's going to have a piece of double-stranded DNA, and then that DNA is going to have to be, or it can be single-stranded, but either way, that DNA is going to have to be a message made from it, so there needs to be transcribed RNA, and then that RNA gets translated into the viral proteins that then rebuild a new sets of nucleic capsids and all those capsimers and everything, and you make a copy of that original DNA piece, and then you have all the parts you need to rebuild. If you're an RNA virus, remember sometimes you come in with the message already. Sometimes you come in with a message, and when you come in with the message, then you need to just make the proteins and make a copy of the RNA. But if you come in with, remember that negative sense, that's when you have to reverse transcribe, so it acts like DNA. And I talked about that in the last video. If there's any confusion, ask me more in detail about that in class, because it's kind of hard to describe without drawing pictures and stuff. But the whole point of synthesis is using the nucleic acid to build more parts. So we're making more nucleic acid, we're building more proteins, so that we can reconstruct a whole bunch of more virons. So then the last step, which is a whole bunch of smoke and mirrors we don't really understand very well, and remember virology is a fairly young science, is how the viruses mature. And this is how they come back and form the new viral particles. Right? For some reason my computer is freezing. Let me just see if it'll flip to the next slide. Okay, so then the last stage is after we have, remember we started with attaching, getting inside, uncoding our DNA or RNA, using the DNA or RNA to make all the parts that you need, rebuilding, and now somehow you've got to get out. So that's the release stage. So a non-enveloped virus, basically what they do is they make so many copies of themselves in the interior of the cell until the cell essentially explodes. And when the cell explodes, all those non-enveloped virons that are inside escape out. If you're an enveloped virus, remember you take some of your host envelope with you. So you undergo something called budding or exocytosis, and you sort of like a bubble comes off with the nucleocapsid on the inside. Now, depending on the type of virus, within one host cell, you can make anywhere from 3,000 to 100,000 new viruses released. And the cycle of this whole process can be anywhere from 8 to 36 hours. So if we look at the budding, okay, so instead of just the exploding, this shows an example of the budding, okay? So you have your capsid here that's starting to rebuild. We have RNA here, whatever nucleic acid you have. And it comes to the surface, that nucleocapsid comes to the surface where there's some viral proteins that have been integrated because you carry the DNA to make those viral proteins. And then they bud off like this, forming, and see how it takes some of that host cell with it, host cell membrane with it. And that's how the envelope viruses occur. And this shows an actual image of that happening. So you can see these are, here you've got the viruses, and they are budding off and taking some of the host membrane with them. Now, in the next video, I'll talk a little bit about some other types of bacterial or viral cells and sort of effects, medically significant effects that the viruses have on host cells.